So one could presume that the use of the animal model here, gauging the effect of sex hormones on HIV infectivity, told us something relevant about human biology. all bans. Can't do any of it. None of it. It's all gone.
This is Audiomunity, a podcast about our body's never-ending fight with the outside world.
Good morning, everyone. My name is Matt Woodruff, and welcome to Audiomunity, episode 39. Our episode today is brought to you by you, our listeners, and also Banned Science. Are you tired of hearing about how your business is negatively impacting the environment? Banned Science. Are you sick of having to hear about how those poor people always seem to be sick? Banned Science. Are you generally tired of people pushing back on your wild assertions about the world? Just ban science. Banning science is an easy and effective way to treat your academic infestations and make sure that you can protect your democracy of one. Because after all, you know best. Joining me today is anger, general snarkiness, and my good friend Kevin Bonham.
I feel like I embody some of those things as well. Hello.
Yeah. Good to talk to you today. So we have a paper that I'm excited to talk to you about, and can you guess why I picked the paper?
I would guess because it contains lots of like really, really triggering ideas, like infectious diseases, like females, like that there are differences between the gender, and it might be important when we do science. It might just be important that we understand differences in gender and gender disparities.
And sex, and the difference between those things, and the impact that a hormone like progesterone has on biology.
Yeah. Well, it's so funny to me. I mean, not to just like jump straight into the politics, although I guess you already did that. So
no,
it's
I did.
Not on me.
Here.
I find it so interesting how much emphasis there is, depending on the point that they want to make, on the idea that sex has a biological component or it doesn't. It's like, can we discriminate using biology? If so, biology, good. If talking about under like a marginalized group, or like talking about the fact that there might be bias somewhere, anytime, in any place, biology bad. Like, we don't want to talk about that. That would be discrimination. No, it's it's quite challenging to navigate the like. But biology says. Anyway.
Yeah. I have to say, so the thing. Like, a lot of stuff has happened. I wasn't here for the last recording, and it's been a while since we...
That recording, we should talk a little bit about it, because people, I don't know if anyone's watching, but if they are, they might notice that I'm in a slightly different location than my typical, because I'm in an office with good internet. I walked across the street, and I have internet. So that's nice. This isn't like a permanent office. I'm just squatting right now. But the last recording Kate and I did, we started it, and things went off the rails, technologically speaking. So we're going to have to re-record that episode.
Okay. So we leapfrogged.
So you weren't actually missing, according to the broader universe.
Yeah. Cool. Well, so I suppose that is why I had my episode numbers all turned around in my head. And so since last time I ranted on Audiomunity, a couple things have happened. There's been a general consensus that no mRNA-based vaccine research is going to be funded on the world. I think I had talked about the possibility of that. We've had a couple grants and things fall apart, which isn't great. But you know what really irritated me a lot, more than I expected, actually, was recent reporting that the Department of Justice reached out to scientific journals to assert that they were worried that there was bias in the review process, and asking them how they ensure that that balanced ideas are heard through the scientific literature, which is literally what the review process is, right? And it is like the thing that they're mad about is the thing that the journal is supposed to do for science. And I know that that's not what they mean when they say that. Like, I'm not dense. What they mean is, I don't like the ideas that are published in your journal. I would like the opportunity, as someone that doesn't know anything about this, to cast doubt on the things that you're presenting as scientific evidence under the guise of fairness of expression. But
no, Matt, I think what they're saying, I mean, the subtext is, I mean, I don't know if you've heard the sort of snark that, like, reality has a liberal bias. The thing that they are asserting is, I want to be able to make up the facts that are politically convenient.
right.
And I don't like the fact that you will not let my facts that I have made up into the pages of your journal. And, like, we've had conversations in the past about my critique of the publishing industry.
Sure.
I do not think I would ever say that there is no bias in the review process. I would never say that the institutions that, as they currently exist, are pristine and perfect. I think there are a lot of problems, and I would frankly really welcome a lot of types of reform. The problem is, these actions are not motivated by a desire to reform. They are motivated by a desire to destroy in a way that allows, you know, a thing that is more politically convenient to be built up in its place. Like, this is also related. I don't know if you saw, I almost brought this up to do on Audio Immunity, but it felt like it was almost too political. The new director of the NIH, Jay Bhattacharya, health economist from Stanford, charitably speaking, has heterodox views with respect to things like whether we should just let everybody get infected with things like COVID-19. He and some of his allies sort of pushed this idea. They sort of, like, launched a new journal, like, out of the NIH specifically to publish, it seems, this one paper about, like, reforms that would be useful to the NIH around, like, granting. And the justification are things like, it's hard for early career researchers to get grants because there's this huge bureaucracy built up around the granting process. And one of the reforms, I mean, they suggested a bunch of reforms, one of which was like, let's just basically fund people, kind of like the way the HHMI does, where they say, like, this is a good scientist, they tend to have good ideas, we're going to fund that person and take away a lot of the overhead of, like, writing specific grants and needing to do all these updates. It's like, if you're doing science, if you're being productive, we're just going to fund you and have, like, different ideas for granting. Like, that, on the surface, that all seems like a great idea. Sort of related tangent, I just recently read this book that's been kind of making the rounds called Abundance by Ezra Klein and somebody else whose name I'm forgetting now. But there's a chapter in there about NIH and about how, like, discoveries are like, the pace of new discovery is slowing down because we've layered on all this bureaucracy and stuff. So, like, there's an argument there I'm sympathetic to. The problem is that we know that these people are pushing this agenda to be basically like, I want to fund another study to show that vaccines cause autism, even though they don't.
Right.
I want to be able to fund a person who I know will come to that conclusion, rather than having them go through, like, you know, meritorious review and all that bullshit. So it's like a lot of things in this environment, it's like there is an underlying truth to some of the critique, but it's being wielded in complete bad faith to try to destroy the institutions
Yeah. While you're making that decision to fund individuals, you're also centralizing the way that review processes are running at the NIH with significantly increased administrative control, right? Like that's
and
that's
by the
the
way,
context. So you control all of it.
Yeah, just by coincidence, merit will probably not include any women or minorities because, obviously, anyone who ascended to any position of authority could not possibly have done that on merit if they are not white men. That's just my opinion. It's not actually my opinion, just to be clear.
And that's the clip that gets out into the world.
Yep.
No, all kinds of problems. So, yes, you are correct. That is why I chose this paper today.
What is the paper, Matt?
The paper is Proinflammatory Oscillations over the Menstrual Cycle Drives Bystander CD4 T cell recruitment and SHIV susceptibility from Vaginal Challenge. And if you tried to write a grant and submit that today to the NIH, I don't think it would even make it. I don't even think it would make it past the initial submission system. I think it would just be bounced right back with a big red nope. Now, the first and last authors on this paper at the time were in the division of HIV/AIDS prevention, and you may have heard of the division of
Let's say who those people
are. Prevention. That is Allison Swames-Kohlmeyer and J. Gerardo Garcia Lerma. You might have heard of the division of HIV/AIDS prevention because that was one of the divisions that was summarily dismissed the other day on April 1st. So everything that I'm about to tell you, fortunately, Allison now is here at Emory. So full disclosure, I hope to work with her on some of this stuff because I think there's some cool B-cell stuff that maybe we'll talk about at some point. But that division no longer exists. So if you were thinking about this paper as we're talking about it and thinking to yourself, oh, that's interesting, I want to know more about that.
That could be worth studying.
Yeah, you can't. Banned.
Yeah, and I just want to say, again, another tangent here, but I have been increasingly frustrated with headlines that say things like, Trump administration cancels, whatever program. Trump administration defunds USAID, etc. Because it's true, but many times these stories are written as if this is like a perfectly normal thing that he obviously has the power to do. It's like I want those headlines to say like, the administration lawlessly dismantles a program at the CDC. Lead testing across the United States.
Right?
Exactly. Like,
OSHA is not a thing anymore. Hope you liked safety in mines. Wear your hard hat.
Presidential administration decides it is the only branch of government.
Yeah.
Right. Anyway.
Cool.
Sorry.
Yep. So now we've already hit the ranting phase, and that's good. So let's we'll walk it back a little bit. We will walk it back to some science
for now.
And we will talk about menstruation for a second. And so
just side note again, I want to acknowledge the awkwardness or strangeness of two white guys talking about menstruation. I think it's perfectly reasonable. This is a scientific thing, but it's one of those things that mansplaining warning is what I'm saying. Just FYI.
Yeah, no, I'm not going to try to talk about anything that I don't understand.
It would be worse if Kate were here and we were like, let's throw to Kate to talk about menstruation.
Yeah,
that would be weird too.
I actually think I'm hopeful that normalizing white male scientists talking about things like this can only be to the good. So, menstruation is the cycle that females go through preparing to have fertilized eggs implant into the uterus so that they then can grow and become children, right? So this cycle in humans follows somewhere around a 14 to 28 day cycle, tends to be pretty regular. Essentially, what we're talking about here
Within large error bars.
Yeah, with large error bars, yep. And what we're talking about here is essentially, like I said, the preparation of the uterus for implantation of a fertilized egg, right? And it requires some really specialized architecture in order to make sure that an egg implants, that a placenta can form, right? That you can create the really, really complicated interface between what starts as a fertilized egg and turns into an embryo, right? So there's an enormous amount of architectural change, vascular change, right? All kinds of things are happening in this space. And we've talked about this before on autoimmunity. There is a very, very large immune component that actually promotes the differentiation of some of the specialized cells that allow for the interface between the placenta and the uterus. It's really, really cool biology and really, really cool immunology. You get into discussions about tolerance because the maternal immune system is exposed to the fetal immune system, right? Like it just turns into this really cool, ornate interplay.
Yeah, and it's complicated in the sense that, like, it turns out, so placental mammals are, this broad group of things. Mice are included in that, but it turns out to be really hard to study because the architecture of a uterus for an animal like mice that can get pregnant very, very frequently, they have large litters, right? Multiple animals compared to the uterus of primates, like humans, it's like very like they're obviously related, but it turns out to be very complicated to study animal models of pregnancy and of the whole sort of reproductive cycle in female animals. So it's like complication upon complication here. Time varying things are hard to study, right? So this is a thing that happens on a particular cycle. Like, there's some methods I want to get into in this paper to sort of look at that. And I just want to highlight two interesting facts we've talked previously about when we talked about this subject on autoimmunity. One is the fact that the reason that we have a placenta is due to a protein that is from an endogenous retrovirus. So a retrovirus that infected some distant ancestor. Some gene got incorporated into the germline, and there's a viral protein called syncytin that is necessary for the structure of the placenta. That's one cool thing. And then the other thing to consider, again, is just like one of the reasons this is so complicated is because fetuses are essentially parasites from the point of view of the immune system. They are foreign bodies, which obviously the immune system is like typically not super happy about incorporating. And so that's one of the reasons the immunology around pregnancy is so sort of neat, and it's an interesting paradigm to study.
Yeah, absolutely it is. And so we are going to get into some of that immunology. We've, like you said, we've talked about this in the past. And when we've talked about this in the past, we've really focused on the immunology of the placenta, right? And sort of the interface between fetal and maternal, which, again, really ornate, tolerogenic, but also kind of reactive in weird ways. Like, it's a very, very cool system. So go back and take a look or find some papers because there's cool biology there. It is rarely studied because, as Kevin just mentioned, it is hard. I would say that one of the things that Allison is doing right now, which is really cool, is putting mouse models together that actually start to replicate that cycling phenomenon. We won't talk about that today so much. But basically, basics of menstruation. So starting from a female that has recently undergone menstruation, right? So you just cleared away the previous month's endometrial lining, okay? So starting from sort of baseline. We have a proliferative phase, which is largely driven by estrogen, right, which sort of re-thickens the endometrial layer. We have a luteal phase or a secretory phase that happens just after ovulation. So progesterone starts to increase, right? The egg is released down the fallopian tubes towards the Wow. Towards the uterus is what I'm looking for. I've said uterus 3 or 4 times. I know what it is. So down the fallopian tubes towards the uterus, progesterone causes that endometrial layer to thicken more and sort of differentiate towards the really sort of nutrient-rich environment that fertilized egg eventually will need to implant and to grow. This is called decidualization. So that transforming of endometrial tissue into this sort of terminally differentiated environment that a fertilized egg can implant in. And so this is going to, we talk about spiral arteries, I think, in the past, and some of the specialized infrastructure that's required for implantation and all of those things. And this is sort of that differentiation process. If implantation happens, then that's great. Everything just sort of proceeds. If there is not implantation, progesterone drops. When progesterone drops, all of that terminally differentiated endometrial tissue starts to die back. You actually get an enormous amount of immune infiltration, which is why I am interested in working on some of this, hopefully in collaboration. There's some cool biology going on there, but eventually that differentiated endometrial tissue dies back and is sort of flushed out of the system through menstruation, right? Flushed out of the system is a really generalized way of thinking about that. It is much more complicated than that. It's much more controlled than that. The immune environment there is very, very interesting, right? So all of this is really complicated, right? You think about, I talk about all the time the changes in immune infrastructure at the lymph node level, right? Like when you're undergoing an active immune response, there are tons of stromal changes that allow you, like germinal center formation, things like this, that allow you to generate a fully fledged immune response. This is wildly, I would argue, more complicated than that. Maybe not more complicated, but on a much, much larger scale than we are generally talking about. When we're talking about secondary lymphoid tissue, there's a whole lot of surface area here. There is a lot of microbiome involvement here, right? They mention in the introduction that there is actually a fair amount of work talking about how at different phases of the menstrual cycle, you are actually susceptible to different pathogens. And it's one of the interesting components leading into this study it was known that there were certain bacterial pathogens, chlamydia, for example, gonorrhea, for example, that are capable of infecting at much higher potency at specific phases of the menstrual cycle, right? And the obvious implication of that is probably the immune components, right? Because they are so intimately involved in this and they are transitioning along with menstrual cycle has some impact on the ability of these bugs to establish themselves and really get going. And go ahead.
Yeah, and part of that is the immune system, and part of it is the changing environment, because there isn't. We've talked before about where is the actual microbiome, and there's some controversy around some spaces. Generally speaking, there's a pretty robust literature on the vaginal microbiome, uterine microbiome. Uterus is probably a bit more exclusive. We don't want to use the term immune privilege, but probably a little bit more like the lung in terms of maybe there's some microbes occasionally that get there, but it's mostly excluded. But the environment of the vagina is constantly changing throughout this period. There's different secretions, different nutrient contents, all of which cause the microbiome itself to cycle. So in terms of susceptibility to infection, you're talking both about immune environment changing. So you think behind the epithelial wall, you have differences, but also on the luminal side of the epithelium, inside the vagina, there's also constantly changing environments, both in terms of microbial presence as well as just the physical structure of the extraepithelial space. So yeah, all of this stuff like, again, vastly complicated and very cool biology that's worth considering. Yep. And I really liked in this paper, I guess this is maybe a feature of this journal. I haven't read a bunch of papers from this journal, eBiomedicine, but there's a nice little box on the second page that says research in context. And so they say like what you would do in a talk where it's like what was known before and what the added value of this study is, and like future directions, like just in a little like nice summary form, which I quite liked. And one thing that it hadn't even occurred to me here is that we are talking about sort of like the base biology of this cycle that you might learn in like a high school health class or something. But of course, these environments are not immune from external intervention. And so one thing they talk about is like hormonal birth control and different kinds of birth control that's used. Those are often hormone-based. Hormones we know have profound effects on the immune system. Some types of hormonal birth control change the periodicity of the cycle, or they change which phases of the cycle actually happen. So like these things are really relevant if we want to talk about susceptibility to infectious disease, particularly for something like HIV, which has an enormous burden, particularly in places of the world that our research institutions no longer care about, apparently.
Yep. Well, I mean, it's fortunate for us then that we couldn't do this work anyway because none of this infrastructure exists anymore. Yeah. So, getting into figure 1.
This is like how it's going to be, people.
Club in the face of sadness. No, I think actually, a serious note aside for a second. I know that people listen to this because immunology is cool. I hope that people listen to this because immunology is cool. I hope that we teach you something. I hope that we sort of keep you on the edge of what people are interested in. It is also, I think, sort of an obligation of ours to make sure that as we talk about science, we also talk about what the impact of that science is on society, where it helps us. I think that is an obligation of scientists in general that sometimes we don't take seriously enough. And so I think that it is actually our job, really, to make sure that you are aware of what is being done out there and also what the limitations of that are and you know what the impacts of changing policy is. So I won't stress any more than that, other than I hope those of you that are listening are not thinking, oh my God, there they go again. Like we really are trying to make sure that you are just aware of what's going on.
And if you value that service, patreon.com/audioimmunity. Go ahead, Matt. Figure 1.
There you go. Okay, so first, does the immune system change through the menstrual cycle? Yes, it does. Figure 1 tells us how. So we're looking at T cells here, basically all T cell biology all through the paper. So the first thing that we're going to take a look at is progesterone. And I mentioned that progesterone is the hormone, the sex hormone. Can't say that anymore. The sex hormone that allows you to go through that endometrial decidualization phase.
Is that related to like deciduous trees? Is that like getting permanent? Is that the...
Got me. Yep. Don't want to answer that question because I don't know. It's like dendritic
I wanted
cells.
to mention too, this is kind of cool. I had forgotten this from, I don't know if I learned this in high school biology, but like the follicle where the egg is released from, basically, like that structure is the thing that secretes the progesterone. Like after the follicle like develops and then the egg is released, then that structure, like as it's breaking down, is the thing that is releasing the progesterone, which is just, it's cool. It's very cool biology.
Well, it's a cool way to make sure that it syncs up, right?
Yeah,
Like you just make the same thing do it. So the egg is released, and you get this release of progesterone, and progesterone allows that endometrial lining to continue to build up. And you can see that in figure 1A. So basically what they're doing is they are breaking down the process of menstruation into 3 phases. the follicular phase, which is sort of that early phase right after menstruation. So you're building up to eventually release the egg. The luteal phase, so this is when the egg has been released and progesterone is building. And then the late luteal phase, which is after the egg has failed to implant, so we do not have a fertilized egg. And the result of that is a drop in progesterone and an eventual release of that endometrial layer through menstruation, right? Or breakdown, at least. And so what they're doing here is they're showing you how progesterone changes over the course of that cycle. And they're also showing you the average CCR5 positive CD4 T cell count in the blood. And we should mention that these are pigtail macaques that we are talking about. And the reason that you use pigtail macaques in this kind of study is exactly what Kevin was talking about before, which is that you can't do this in mice effectively because the cycling looks a lot different. The entire infrastructure really looks a lot different. Large litters, as Kevin mentioned, are a thing. So it's just hard to do this kind of study. And another plug, when you hear the next NIH push towards non-animal-based research in favor of organoids and synthetic biology, good luck to you doing anything related to women's health in organoid or synthetic biology research, because there's simply not enough known out there, because no one was studying it before, and they're certainly not studying it now.
I mean, yeah.
Yeah, boy, the level of snark today is high.
Organoids are freaking cool.
Yeah, they are.
There is a lot of very cool synthetic biology. Mm-hmm. We are somewhat famously not able to reproduce uteruses in vitro.
Yeah.
Would change a lot of things if we could have a synthetic uterus. We definitely don't have that.
All right. So we are looking at basic biology of how progesterone is changing, cycling, and it seems like CD4 T cells are also changing over the course of that, right? So as progesterone goes up, CCR5 positive CD4 T cells are also going up, and so these are cells that are indicative of a type 1 inflammatory response. And so basically what we're doing here is we're just tracking levels of progesterone, sort of defining what we believe to be the follicular phase, the luteal phase, and the late follicular or late luteal phase, and then asking the question, how are these T cells changing over that time? And the answer is they're changing in a number of ways. So in D, we've now gated on CD4 T cells, and they're showing that from the follicular phase to the late luteal phase, you've got an increase in CCR5, so an increase in these type 1 inflammatory T cell responses. Similarly, an increase in PD-1, and a decrease in FOXP3 expression, ish. And Kevin's making a face because, like, maybe, boy, the stats look good. And I would argue, to be fair, they hammered themselves by trying to plot all these things on the same scale.
Yeah, but even still, I mean, the error bars are overlapping the means. I
get it.
The stats look fine.
Yep.
Everything is in the same direction. This is a place where trend, I think, is a valid thing to note. Everything is sort of like in the same direction. Yeah. I think if you did false discovery rate correction here, it would just destroy many of these results. And like the effect sizes, when you're talking about stats, there's two pieces. There's like the thing that we use to judge significance, like statistical significance. Those numbers all look fine. Effect size is like, how big is the difference? For many of these, I mean, the PD-1 looks pretty good to me.
Yep.
Some of the cytokines look okay. Some of these, like, they have marginal p-values, and the effect sizes are also a bit small.
Yeah.
But again, it's all trending in the same direction, and I think the later results sort of like back this up. But it's a little.
I would say in aggregate, without going into details in much of this. In aggregate, the there is seemingly a fluctuation in T cell biology. There seems to be a generally increased level of inflammatory type cellular response or circulating in the blood, and probably also a general decrease in regulatory function through that FOXP3. Now they look at the CD4s that are expressing in relation to average CD4 expression. This is actually an interesting way of plotting these data over time. I was not familiar with this, and I may actually incorporate this because it is hard to decide what you're going to compare to at any given moment, and I think a running mean is not bad here.
Yeah, and I should also say, like, to their credit, like, we've talked, like, this is becoming a theme of season 2 of Autoimmunity. This, like, when you're doing this, like, these pigtail macaques are not genetically inbred. There's lots of diversity. You can think in most studies of this kind, people are taking blood like whenever. Like, most people don't record the menstrual cycle of the female animals, right? Many studies don't even include female animals, specifically because it's complicated when you have these things that can vary by time, and like the effect size. This effect size of the phase of the menstrual cycle is larger than the effect size of some things that people are recording, like that are other kinds of biology. So you can imagine, like, in a study where you're not paying attention to menstrual cycle, it matters when you're drawing those blood, right? Like those blood samples.
Sure does.
And most people just don't consider it, or they only do male animals specifically for this reason, because they want their error bars to be tighter. And it's challenging to do biology when you have this, like, weeks long oscillation in many of these phenomena. So,
but you know how you deal with that? Ban it.
So, yes.
If you so we have changes in T cell biology over the course of the menstrual cycle. The broader question, I think, becomes: is it just T cells? And the answer is seemingly no. Now, all that diversity that you're talking about here, and I can tell you, Kevin and I have had sort of a side project running that we're excited about diversity outbred mice. And
don't use that word, Matt.
I know. And how immune systems can change. And I can tell you that even if you just take mice that have been inbred and start to put them into an outbred background, there are enormous changes in circulating biology that you see. And so you can imagine that that's reflected. It certainly is reflected in humans. It's also reflected in pigtail macaques. And so what they're doing is basically using spectral flow cytometry based setup to break down their circulating blood populations into major immune subsets. I would say that the sample sizes that they're working with here are a little underpowered, it looks like, for most of this data. There are some clear things that show up. They've got changes in monocytes, I would argue. So CD4 positive, CD16 negative monocytes seem to be decreased in the late luteal phase, which is interesting. Maybe there's a change in conventional dendritic cells, although those are really a crapshoot in the blood anyway. Those are really tissue resident, so it's always kind of hard to interpret what those mean. Interestingly, the one thing that does seem to very clearly change is CD40 positive B cells. So,
not that Matt has any particular interest in that cell population,
but it's fine. So those, to be clear, would be B cells that are capable of interacting, for example, with a T cell and presenting antigen. Who's to say what those B cells are doing? But
I wanted to just highlight here a quick method thing, which is that they're using here to do the modeling to get the statistics out. They're doing something called a generalized estimating equation, which I had not previously been familiar with. But this is basically a kind of linear model that is working to. One of the problems with this kind of study, when you have these oscillations, is that you have to do repeat measures of the same animal. And it turns out that in many cases, the best the best estimate of what a particular value is going to be at time x is what it was at time x minus 1, right? So like if you measured a particular T cell population at one time, it's going to be heavily correlated with the next time you measure in that same animal. So a lot of the assumptions that statistical models often have about independence of observations are violated when you're doing repeat measures. So there's a couple of different ways of dealing with this. I would say more typical, or I've seen it more frequently, is something called a generalized linear model, where you are including a term in the equation that is the individual identifier to do something called a mixed effects model. This GEE is essentially, it's a kind of mixed effects model, or it's related, but at least what I read on Wikipedia is that it can sometimes have more power when you are dealing with lots of repeat measures. It sort of deals better with that autocorrelation piece.
Yep.
For math reasons that I didn't look into, and frankly probably wouldn't understand even if I did. So
and there you go. That's the end of our expertise. So there are cells that change along this cycling. And also, it would be interesting to know if inflammatory cytokines change, right? Because these things are all obviously responsive to inflammatory signaling. Maybe you can read that out. And the answer is kinda. So if you take a panel of inflammatory cytokines and you generally assess for a bunch of them, so IL-1 beta, IL-6, MCP-1, IL-8, TNF-alpha, interferon gamma, and then you ask the question, are any of those individually changing? And the answer seems to be maybe. If you take all of those things and you add them together as a sort of generalized inflammatory sort of thing, the answer is probably. It's again, it trends with this previous idea that, you've got clear changes in T cell biology that seems to correlate with progesterone, and that seems to correlate with generalized changes in systemic inflammatory signaling, right?
I feel like, not to like toot my own horn here or anything, but I feel like this study is a perfect candidate for doing the kinds of things that we did in our COVID paper, which is like use a machine learning model that can handle high dimensionality and sort of like bring many features together. Because when you're doing this feature-wise testing, it's obviously like it's very robust when it works, but in this kind of situation where you've got so much variation, I feel like there are machine learning models that could handle, and this even isn't even like hundreds of features, right? Like we're talking about a couple dozen. They've got the sample numbers to, I think, make a probably pretty robust predictor here if we threw this into a machine learning context.
Yeah. Yep. Agreed. We could pitch that. So.
Not that this work is getting funded, so.
Right. Nonetheless, I will be doing the flow. So, cool. So
we're not bitter.
It is then interesting to know if you are interested in HIV, for example, which these folks are, because again, this is the CDC division of HIV prevention.
Was the division.
Yeah, thank you. You would want to know if these changes change the potential susceptibility of HIV. We know that changes in cycle or differences in cycle are going to have an impact on things like gonorrhea. Is that the same in HIV? And so we are going back and we are asking the question, are the target cells of HIV in the vaginal tract different over the course of this? And you might be asking yourself, why are we talking about CCR5 and type 1 inflammatory signaling and all of that in the top of this paper? And I would remind you, if you're asking that question, that CCR5 is a critical co-receptor in HIV binding to the cell surface. And so a CCR5 positive CD4 T cell represents a nice juicy target for a new HIV virion introduced into the system. So one might be concerned that if CCR5 is going up in the blood on T cells, that perhaps that's true in the vaginal tract as well. And one could imagine that that would leave you more susceptible in those moments to HIV transmission. And so the first question is, of course, do those cells show up in the vaginal tract? And the answer is yes. The answer is through longitudinal vaginal lavages alongside the blood collections that they were doing before. You can basically go through and you can ask, are there increased T cells in the vagina, in the vaginal tract, in the female reproductive tract in general? FRT, they refer to it as. In the late luteal phase versus the follicular phase. And there's an increase in T cells in general. There is, go ahead, sorry.
I was just going to say, this is a question I had here, and I don't know if you know the answer, but I was actually sort of surprised that this technique works to see T cells. Like, normally I think of things like T cells as being, on the basolateral surface of any epithelial surface, right? Like on the inside, essentially. Like there's a layer of epithelium, and then the T cells and dendritic cells and all of those things are going to be behind that layer. And a lavage here is just like we're essentially just like moving fluid through the vagina and then like collecting it on the other end, or not, on the same end rather. We put water in, or like saline in, and then we take whatever comes out. So like obviously there's going to be some epithelial sloughing and things like that. But like I was saying at the beginning, like the actual environment of the vagina is so different at these different phases. I was wondering if we think that it's similar sampling, as opposed to obviously doing this longitudinally with if you're like sacking the animal and taking out tissue would be that's a much more expensive proposition. You've got to actually kill the animals, or even doing biopsies or something like that is much more complicated. But I'm wondering if this lavage, if we think that is, if the numbers we're getting out of that are an accurate reflection of tissue residency for these cells.
Yeah, I think it's a very relevant question. And, this is not my area of expertise, obviously, but if we're talking about an endometrial layer that is in the process of being sloughed off, right, it is essentially detaching anyway, and there are T cells in that, and by lavaging at that time, you are simply helping the process and washing it away, and so you're getting increased sampling. I think that is a completely reasonable thing to ask. And I mean, there's a dramatic shift, right? From
these effect sizes are not small.
in L, you're asking the question, the cumulative number of HIV target cells in the late luteal versus the follicular, and the answer goes from basically none to hundreds or thousands. And nope, I think that is completely relevant. Now, I think that a biopsy would tell you much more. I think that imaging would tell you much more, and I can tell you that there is some imaging over in the lab that has sort of grown out of this a little bit that is also very interesting. But yes, I share that concern here. But nonetheless, certainly target cells.
All trending again in the same.
Yep. Certainly trending in the right, in the direction that the authors are intending. A question becomes, are we really in sync with our assertion that CCR5 and progesterone are truly cycling together? Do we have a direct correlation? So figure 4, I won't spend a lot of time in, but basically what they're doing is they're just going back to the same animal over and over and over again through, a good between 2 and 6 cycles, it seems, and essentially just showing that as progesterone goes up and down and up and down and up and down, CCR5 positive T cells go up and down and up and down and up and down, right? So these two things seem locked.
This I found very, very convincing, and this is the modeling here again is kind of interesting. There, I think if I'm not mistaken, this is 5 different animals, and we're seeing a plot for each animal individually, because what they're effectively doing is they're trying to fit effectively a sine curve, but of course it's not going to be as uniform as a sine curve. It's going to be a complicated thing, like not every period cycle is exactly the same periodicity. The amplitudes are different, and so they use this model, this time varying model, which I wasn't previously familiar with. I'm more familiar with things like Gaussian process models for this kind of thing, but basically you are trying to fit an oscillation and get stats about that oscillation in a situation where you are not sampling at a super uniform rate, and all of the parameters of the oscillation can change from cycle to cycle, which is complicated, as you might imagine. They're actually using techniques here from digital signal processing, which I'm not very familiar with. I did a little bit of reading, but so this Lomb-Scargle power model, which is like trying to fit. I know it sounds.
I can't say that seriously. Nope.
Yeah, it's
somebody's name. Have some respect.
Somebody's
Kevin.
name. It feels like a growth that you would pick off your Lomb-Scargle, but this model is basically trying to understand that periodicity and gets kind of summary statistics over multiple iterations of this non-uniform periodic events. So, but if you just look at the plots, I mean, that model aside, if you just look at the levels of progesterone week to week and levels of CCR5 week to week, it's just it looks very clear.
Yeah.
It feels like you almost don't need the model. It's nice to have the model, but you almost don't need it. It's just I thought that part was very convincing.
Yeah, quantitation's good generally. So this is where I really like this. This is actually a very cool experiment. Figure 5 to me makes the paper. So basically, pigtail macaques are susceptible to simian HIV, right? You can imagine that if you give a low enough inoculation of simian HIV into the vaginal tract, you will get low levels of infectivity. So one might imagine if there is cycling of the immune system during menstrual cycle, and if that has an impact on the number of target cells in the vaginal tract, then you might expect that you could be more susceptible to HIV at those specific times, right? So basically what they do is they take these macaques and they are looking at, I think it is, what is it? How many? 6? I want to say 6. But basically what they're doing is they are inoculating these female macaques at regular time points, and they're also collecting blood at regular time points, essentially. Now, they don't know when those females are going to get infected. So what they're doing is they're basically collecting at relatively frequent time points. They're waiting until you get a change in viremia, and when there's a change in viremia, they are backtracking 7 days so that they can identify the time point at which the infection occurred. And then they're using that as time point of infection and carrying forward. And they're looking at what was the blood situation, right? What markers were there at the time point of infection, and does that correlate at all with the menstrual cycle as previously defined in the rest of the paper, right? Which is cool. That's hard science to do, especially in macaques. Like this is a hard, cool experiment. I really like it.
Just to put a fine point on it, like the challenge, the SIV challenges are less than 100%. That they're going to lead to productive infection. So this is I don't know if it's a dose thing or if it's just the particular infectivity of this virus, but they're challenges, and they show actually in A, this was actually quite helpful. It took me a sec to be like, why are they doing male mice or male macaques all of a sudden? But so time point A, or panel A rather, is for male animals which presumably are not undergoing menstrual cycles. they are doing the same frequency of challenge with the virus to show how long it takes for it takes at some point after some period of time all of the animals are going to be infected, but the specific frequency of when those infections happen for a male presumably would be uniform, the particular probability, but for the females they want to know, is it changing depending on the menstrual cycle?
Yeah. And the answer, to cut straight to the chase, is yes, it does matter. So what you can see is after that first time point post-infection, about 3 weeks later, there is a crash in CD4 T cell counts that correlates with viremia spiking. This is all stuff that is known in terms of normal HIV infection. But then what you can do is we can go back and ask, what was the T cell biology in the blood at the time of infection? And so we've previously talked about CCR5 positive T cells increased in circulation in the late luteal phase, the PD-1 expression increasing, FOXP3 expression decreasing, TNF-alpha expression increasing, right? In every single one of those scenarios, it turns out that the time point of infection, if you look at the blood signature when those animals were infected, it is much more similar and actually even exaggerated over the late luteal phase versus the follicular phase. So you're basically phenocopying an expanded late luteal phase as the time point of infection every single time, right? Which is super interesting, right? So strongly suggesting that the periodicity of the menstrual cycle is having a major effect on when these animals are actually getting infected.
Yeah. We should say that it's, this is a small number of animals, so it is not the case that you can't get infected except in the late luteal phase. We want to be clear on that.
Yeah.
But yeah, it is. It adds a ton of strength to the idea that this matters.
Yeah.
In terms of susceptibility.
Yeah. So by the end of this figure, and they sort of form a composite, a susceptibility composite at the end, and show you that essentially, whatever makes up that composite, your time point of infection is going to be very similar to what you see in an extreme version of the late luteal phase.
Yeah.
Which I think is really compelling and helps defend all of the stuff sort of up until that point. I think you've just shown that there is actually a very real biologic impact of infection susceptibility. So I really appreciated Figure 5. I think it's cool. I think, that's one of those things where had you just published that, it would stand on its own. Obviously, you publish everything up that to defend Figure 5. I get that, but that's the figure in grad school that we would have pulled out.
This experiment matters more than any of the stats or effect sizes in previous stuff.
Yeah, totally agree. And then Figure 6 is the mandatory, so then we did it in humans, obviously not infecting humans with HIV, but basically
is that
asking...
obvious? A lot of people in the current administration are interested in challenge trials.
Yeah, don't do that with HIV, please. That's when USAID will get kicked back on, because we'll do those in El Salvador.
We're running to the end of time, so I shouldn't keep doing this.
So we are in humans now, and asking the question, if we also track progesterone in humans, do we see the same influences of progesterone on T cell biology circulating? The answer is pretty much.
Yeah.
It seems relevant. I mean, there's a reason this model was chosen in macaques, but seems relevant to humans too. So one could presume that the use of the animal model here, gauging the effect of sex hormones on HIV infectivity, told us something relevant about human biology. all bans. Can't do any of it. None of it. It's all gone. Cool study.
Very cool.
Yeah.
I'm excited that you're collaborating with this person.
Yeah,
seems cool.
I'm excited too. Yeah, and you know how these things go. We'll
Get me in
do
on
some
that.
Cool preliminary, hopefully some cool preliminary stuff, and see where it goes. But nice. I mean, this is the kind of immunology where so little is done.
Yeah.
And it has such importance on sort of general human populations that, it's kind of a gift to meet collaborators like this, because you get the sense that like literally anything that you do, anything that you find becomes highly relevant to an enormous portion of the human population in a meaningful way, which is why
and for
the frustration is so high around this.
For anyone that is thinking like, well, like HIV isn't as big a problem in the United States, like maybe this is something that, foreign governments should care more about than us. I would just say that like this is one model where there's an obvious effect in the vaginal canal, but like the effects of immune cycling they're true throughout the entire body. Like hormonal differences in immune posture are happening systemically. So like, okay, that affects half the population of this country. And then also like, this isn't the only oscillation, right? It's like a monthly oscillation is a big one, but there are changes over time based on circadian rhythms. There are changes that may affect on different cycles depending on things like diet and so this kind of thing, like this is one model, but all of this kind of biology has broader relevance beyond the HIV context, beyond the specific menstrual cycle contents. Like all of this biology like has broader relevance, and this is one of the reasons why we need to fund not only like I have been a little bit like I think it makes sense when we're talking about all of these cancellations to like focus on things like cancer funding and but I think we often forget about the fact that a lot of basic science has relevance that we don't yet know, because all of this stuff is connected and complicated, and like we need the larger picture. So
yeah, and I would also say, just before we go, that the person that is theoretically calculating that maybe this isn't such a big deal in the United States has baked into their understanding a functional division of HIV prevention from the CDC, which was a world's leader for the last 40 years. So when you're thinking about that, what you're really thinking of is, boy, I hope that I can estimate the impact that the CDC has had on HIV in the United States as 0, right? Which is not true.
No.
Yep, we're going to see the effect of that. So,
This has been Audio Immunity, episode 39. I'm Kevin Bonham with Matt Woodruff. This has been a raucous episode, but hopefully people enjoyed it. And if you enjoy what we're doing here, you can like and subscribe to the show on your podcatcher of choice. You can find us on the web at audiomunity.org, AUDIOMMUNITY.org. You can find us on Patreon, patreon.com/audioimmunity, if you want to kick us a little support every month. $1 or $2 a month would go a long way. And
music at the beginning and the end.
Yes, thank you. And the music at the beginning and the end of the show was composed by Rachel Rannick. We'll see you next time.
See you, everybody.
All right, and I need to run.
Yeah, so, right on time. Talk to you later.
99% uploaded already.
There you go. Good deal. All right.
Sometimes you get lucky.